But isn't a drug safe when it gets to the market? But isn't a drug safe when it gets to the market? This is the question that you will be able to answer at the end of this lesson. Drug development is divided in two phases, a preclinical and a clinical phase. During preclinical development, a compound is identified and some drug design takes place. In the lab, some basic efficacy and toxicity studies are done, sometimes in vitro, sometimes on tissue or in the animal model. If these results look promising, the first dose is given to man, and this is the start of clinical development. Clinical development, again, is divided in three phases. In phase 1, we perform pharmacokinetic studies. We administer the drug to young healthy volunteers, sometimes between 20, at the most 100. If the drug looks promising and no major concerns are identified, we can enter phase 2. In phase 2, we investigate proof of concept and dose finding. Here, the participants of the studies will be patients that are strictly selected but have the target disease. We will expose maybe a few hundred patients at the most. Again, if our drug takes this hurdle, we will go into stage 3, where a much larger exposition takes place. We will have now something between 300 to a few thousand patients with a target disease. Against, they will be highly selected. And what do we mean by highly selected? Well, if we want to prove efficacy of a drug, we need to get rid of all the potential confounders. Confounders are factors that might obscure our results or introduce a bias. So, if possible, we try to eliminate patients that have other diseases. We will not take patients into studies that take other medications. And for safety reasons, we will not expose people at the extremes of age. We will exclude children or the elderly from our studies, unless our medication is being developed for use in a pediatric or in a geriatric population. We will also exclude pregnant women, and depending on the characteristics of a substance, we might go so far as to exclude any woman of childbearing age. On top of that, studies are limited in time for financial reasons, even though a drug might be developed for chronic use. So, at the time of submission for marketing approval, the Drug Regulatory Authority will have data only on about 3,500 patients. This means that due to the small sample size, the highly selected population, and the limited duration of a clinical trial, only very common adverse effects will have been identified at this stage.